So hello everyone. I'm very happy to be here to present our project today. It's good that we can do that. So I am Evarist Planet and I come from EPFL, from Life Sciences, and more specifically from the lab from Professor Didier Trono. And this is a collaborative project that started with the SDSC a year ago, more concretely with the help of Luis, which did a terrific job, not only with the analysis, but also to understand our needs, which is probably more difficult. And as you can see, it's named TE4med. It's transcriptome-based identifier for precision medicine. So I will start explaining you the problem we want to tackle. That's what we, in cancer medicine, we call CUPS. So CUPS, it's what's known as cancers of unknown primary. This is between 3 and 5% of all cancers that are diagnosed. And what makes them special is that it's a kind of a diagnosis. We know that it's metastasis, but we don't know where this metastasis was originated. So we don't know which tissue was the starting point of this metastasis. The patients that have this disease are, unfortunately, they have a very bad outcome. They have a median survival of between 9 and 12 months. And as you can imagine, knowing the real site of origin of the cancer would help a lot in treating them. So to better understand this process, if let's assume we have lung cells that become cancerous, at some stage of the cancer evolution, these cells, lung cancer cells will enter the blood vessels and move through the blood vessels to another side of the body, and there keep growing, creating this metastatic tumor. One thing that's important to remember is that these cells that are at a different site, they keep being lung cells from the beginning. It's just cancer lung cells that move to another place. So they keep having lots of markers from the original tissue. So that's something that we can use to trace it back. And in general, in cancer, therapy and everything is directed by the starting tissue where the cancer has been developed. So, a bit more detail about the CUPS. Many diagnostic workups are being used on them, and unfortunately, mostly they fail to detect the tumor origin. So then it deals us with the treatment we give to patients. These patients have a poor risk. They are considered poor risk patients, and therefore they get palliative treatment mostly. So that's chemotherapy, where the specific drug that's given is defined by the subset of CUP that we are treating. In this subset of CUP, the doctor finds it out just looking at the histology of the cells to see if he can find general differences between them. Then some other factors that influence the prognosis are the age of the people, the performance, so how well they respond to the drugs, comorbidities, this is other diseases they have on top of the cancer, and disease extension, which is if the metastasis has been found in many other body sites, and basically how much it has been spread. So you can imagine that the main aim of the project is given a certain tumor sample to find out exactly which the site of origin was of the cancer. For that, many groups have been working on that before. We believe we can build on top of that because we have several tools at our hand that others didn't have at the time. So the first one is transposable elements. This is the specialty in our lab. We work on these so-called also jumping genes, therefore the cartoon. Then we also have a lot of big data at hand to work with, and we can make use of machine learning tools that comes with the help of the Swiss Data Science Center, this part here. So I will walk you through these different tools to try to explain. So transposable elements are around 45% of the genome, and this is something that's under discussion because many people think that it's between 60–80%. That depends on how easy we can detect them, since the detection is sequence-based, and this sequence decays over time, so it's every time more difficult to find the oldest ones, the ones that were all inserted in our genome more time ago. But in any case, this is a largely unused source of our genome in terms of analysis. The reason why it's not used is because it's more difficult to work with than to work with genes or other regions. That's due to the sequence of them being very repetitive. So different transposable elements in our genome have the exact same sequence. Yet, it has proven to be a high-density barcode for cell identity, differentiation, and stage and activation status, which already gives hints that they can help well to identify the site of origin. And more specifically in oncology, we have seen them doing a great job to stage tumors, so to find out in which phase of the disease patients are, to evaluate the clinical prognosis, to find out if someone has a better or worse outcome forecast. And lately we have found out also that they are very good at predicting response to drugs. So the fact of having certain of these transposable elements expressed tells us if specific drugs will work or will not work on patients. And the last point, to evaluate novel therapeutic avenues, this is more a thought we have that it might help in that. So that was our first tool, was the transposable elements. Our second tool is the big data. We are lucky to have access to this data set, the gene tissue expression data set, which is coined in the field as the GTEC. It's RNA-seq data. It's from around 9,000 samples from 500 deceased donors. They claim they are healthy donors, even though they are all dead, but I guess they are healthy dead. And for them, we have this RNA-seq from 54 body sites. So obviously, since it's a RNA-seq, we are talking about expression data here. So we have expression data for every sample for around 20,000 genes in the genome and more than 4 million transposable elements. So this is the causes of death of the people that enter the study. In the darker color, we have the older people, and in lighter blue, we have the younger. So we can see the older people die mostly from heart disease or liver or renal problems, whereas the youngest, it's mostly a traumatic injury, which is car and motorcycle accidents and these kind of things. So at the end of the day, what we get from this is a big table where our cases are samples, these 9,000 samples, and our features are the genes and the transposable elements. It's 4 million of them. This table being flowed, it's around 160 gigabytes of data. So we saw the first two tools, which was the transposable elements and big data. Now we come to the machine learning part. This is where the Swiss Data Science Center jumps in. At the beginning, we saw that every tissue, we believe it will overexpress some specific features, name it genes or Ts. Like, for instance, the stomach will overexpress some of them, and the intestines will change their expression in some others of them, and so on and so forth until we have the full tissues of the body. And additionally, there will be other features that will change their expression depending on other factors, like the gender, the ethnicity, and other things like that. So what the SDSC proposed to model all this endeavor is to use latent variable models. So with latent variable models, the nice thing is that we can capture these underlying latent components of the data. If we look at it the other way around and we look at the different tissues, every one of the tissues will be represented by one or more latent variables, and each of them will contain the different features that we saw before. In this example here, we can see how the first latent variable would be, for instance, encoding for the brain, and we would have genes A and B, and the second one would be stomach, and the third one the intestine, with some genes in common. We see D and E in both of them, so things that could, this is just a cartoon, but we expect to see something like that, with some genes specifying the hierarchy that we have in tissues, whereas we have other genes that are more specific from every site. So in the end, from an unlabeled data set, we can come up with a hypothesis of how the data has been generated. This is, in other words, it's just a simple discomposition of the data. we just try to find, by modeling, the decomposition that better explains our data. So we have our matrix X, which is the expression, where we have, here's an example, where we have rows as samples, and the columns would be the features. We can see that we have, for instance, 3 brains and 3 stomachs there, and we can decompose that into 2 matrices, these matrices Z and B, where Z encodes for the different latent variables. We can see how the first column, for instance, would be telling us which samples are brain, and the second latent variable would tell us for the stomach. And then matrix B could be interpreted as the different weights that each gene has over the different features. So this is a nice approach, but as you might have realized, it has one problem, that is, how do you set the number of latent variables there? We could choose to say, well, as many as the tissues, but then we could lose some granularity, or basically we could put too many. So a good solution for that is to use general latent feature models, which is also known as GLFM. This is a technique that's very nice to infer these latent variable models, and it has the nice property that it does not require to fix a specific value for k. The model will learn this k value from the data, meaning that if we keep adding data to our model, at some point he might decide that certain tissues better explained as 2 of them, or the other way around, that 2 groups of samples can maybe be better explained if put together. So this will bring us to a model where we have all the different tissues and possibly, or other different latent variables encoding for other variables that can be of interest. Okay, so now that we looked at the 3 different tools that we are using, I can show you some of the performance of the method. In this case, Luis used the tissues where we had more samples. So he removed some of the tissues where we had less samples. That's why we have 8,000 samples. And he used just the genes without the transposable elements here. This is a TSNE representation where we can see the different clusters of how the different tissues can be well separated and classified. There are some that are put together, like the breast and the blood vessels and things like that. It kind of makes sense since the breast is full of blood vessels, for instance. And I would say it's a rather nice separation. You can get it also with more of the features, but the nice thing, I believe, is that he's just using 2,000 and something features in this first approach, which is, gives a pretty nice result already. Then, obvious question of this is to look at what's the error rate, or how much misclassification we would have with our model. This is what we try to plot here. It's the misclusterization rate. In the y-axis, we have the error rate, so the percentage of samples that we would classify wrong. And in the x-axis, it's the number of features that are being used in each model. So every dot would be a model. We have this tumor on the right, the red and blues, which are not using the latent variables. They are just using the most expressed features. We can see how they don't reach a very good error rate. They are at 7–8% and using tens of thousands of features. Whereas with the latent variable models, we much faster come down. And between the ones that are using the latent variable models, the one that's working better is the one that's using the genes and the Ts, which reaches an error rate of around 3% with just between, what is that, between 500 and 1,000 features. So there we're pretty happy with that. Still, there is many things to do, but let me go through the conclusions. So this GLFM proves valuable to explore the data already. It's very informative, the kind of features we obtain that are representative for every tissue. They were learning a lot through that on the lab. We learned also that we can reduce the set of features probably just by using the most interesting ones. So most likely we'll have to do some kind of feature selection to reduce dimensionality also. And it has also allowed us to capture relations between features of different tissues by having by having features in different latent variables that are explaining different tissues, we can learn a lot about how these guys interact between them. We can also see that the latent variables might encode new traits that we didn't think of, since they are learned from the data. And they also capture relations between the genes and the Ts, which also give us a lot of information about the regulatory networks between them. Then, this is the stuff we have still to do. There is a lot of job to do to look at the results that we already got from the SDSC and from Luis. And one part that we are working, well, that the SDSC is working intensively now is to improve the computational part of the method. Because right now the method takes like around a week to run, and just on a subset of the data. So they are actively working in extending the algorithm to make use of the sparsity of the data and allowing for higher dimensions. So we will need to scale it up. Another problem we have is that the data we are using is not cancer data, it's healthy tissues in principle. And of course we want to work with the cancer samples. So this difference between the normals and the cancer samples, we will try to break this distance using yet another data set, which is the Cancer Genome Atlas, which is of a bit bigger than the GTEC. The GTEC is around 9,000 samples. This would be around 14,000 samples, sorry. And we will have to add that layer also to the current model. We would also like to add some knowledge that we have on the data. So we know there is structure in the genes. We know that there is distal relations between them and between the transposable elements. And we also think it's very important to add the prior knowledge about metastasis. For instance, we know that breast cancer tends to produce metastasis on the bone. So if we have a metastatic sample from the bone, the breast is a place that we should look at more carefully, probably. We should put weights on it. And finally, we need to validate all this. And for that, we have yet another data set that's of 500 metastases from which we know the origin, where the cancer was originated, so that we will also run through and check the error rate again there. And yeah, with that, I'd like to thank you for your attention.